Please take your seats, everyone. I'm calling this session to order. Good morning, everyone. I'm Jeanette Wing, the Executive Vice President for Research and Professor of Computer Science at Columbia University. I have the honor of moderating this panel on AI and scientific discovery. We will hear from experts on how AI, especially with recent advances in generative AI, can help scientists make new, creative, and surprising discoveries in new, creative, and surprising ways. The panel has 4 experts. I'm going to speak for 15 minutes. Then I'll introduce each of the panelists one by one. Each of them will speak for 15 minutes, and then we will entertain questions from the audience. My talk has 3 parts: a compressed history of AI, a simplified view of transformer models, which underlie generative AI, and a bird's eye view of how AI can benefit science. Putting AI in context. In 1950, Alan Turing, considered the father of modern computer science, proposed the Turing test. If a human interacts with a machine, and the human cannot tell the difference between interacting with the machine and interacting with another human being, then the machine passes the Turing test. We could thus consider this machine as exhibiting, in some sense, human intelligence. 1956 marks not just when the term artificial intelligence was born, but the start of AI as an academic pursuit. At a conference held in Dartmouth, the participants asked whether we could build a machine that mimics the behavior of humans. This grand goal of AI was recognized early on as way too difficult to achieve, and thus subfields of AI splintered off, representing subtasks of human intelligence: computer vision for vision, speech recognition and natural language processing for language, robotics for mobility and manipulation. The first wave of AI during the last half of the last century is signified by representing knowledge symbolically, not numerically, and representing reasoning by rules. For example, if Socrates is a man, then with this rule I can conclude Socrates is mortal. These rule-based methods led to what are called expert systems, which by the late 90s found their way into scientific domains. The first expert system, Dendral, captured knowledge about organic chemistry and let chemists identify unknown organic molecules by analyzing their mass spectra. The second wave in AI hit by the end of the last century, and we are still riding it. The second wave is distinct from the first wave in that it is driven by the plethora of digital data, especially data that represents human behavior. For example, what our movie preferences are, when and how we commute to work every day, and what groceries we buy. By 2012, deep neural networks showed how, with big data and big compute, machines could perform tasks as well as or better than humans. For example, suppose this deep neural network were trained to classify images. Then if I feed it this picture that it has never seen before, it will output that it's a cat. More precisely, it might tell me with high probability, like 0.99, that it's a cat, and with a low probability it's something else, like a tiger. Fast forward 10 years, we are now in the age of generative AI, where we can generate new data. For example, text that has never been written before, or images that have never been created before. Here, the large language model GPT-4 is generating a proof requiring knowledge of calculus to a simplification of a problem statement that appeared in the 2022 International Mathematics Olympiad. In the second example, a diffusion model is used to generate new images of a dog that are similar to the original dog image. It does so by first adding noise to the original picture and then denoising to get a brand new image. To put science in the context of this AI timeline, by the 1980s, supercomputers became the workhorse of science. For example, performing enormous numerical calculations and running simulations of physics-based models. The explosion of scientific data generated by devices and instruments that make observations and take measurements enabled the age of data-driven science. At about the same time, statistical machine learning pulled the splintered subfields of AI back together, bringing vision, language, and robotics closer to each other, and even teasing us about the eventuality of artificial general intelligence. For science, the big breakthrough event came in 2018 with AlphaFold, an AI system built on both deep learning and reinforcement learning that could predict protein structure. Our first speaker, Pushmi Kohli, will talk much more about AlphaFold. It's the convergence of big compute, big data, and advanced AI that provides a context of today's panel. In just the past 10 years, the second wave has turned into a tsunami. Here we see a 3.5-month doubling in the past 10 years in computational power, measured in petaflops per day, used to create machine learning models. Note the y-axis here is a log scale. And in contrast, the 2-year doubling of Moore's law looks like the lower part of this curve. And here we see a 2.2 times growth rate in training data size. In just the past 4 years, we have gone from talking about billions of words to trillions of words. So how can scientists ride this tsunami? It's worth understanding a few basic concepts underlying generative AI. For example, to generate text, if I feed a large language model a sequence of words, then the large language model will predict the next word. That's all it does. It will produce the word with the highest probability of occurring next. For example, if I feed in the input sequence, the cute dog begged for a, it will output bone, assuming no other word has a higher associated probability. As mentioned earlier, I can generate new images using not just LLMs, but diffusion models. What is fundamental to both techniques is that the underlying probability distributions are learned from billions of examples and represented as deep neural networks. In the next few slides, I want to sketch out the transformer architecture, which is shared by state-of-the-art large language models. The transformer architecture has 2 steps, and both steps build on DNNs. In the first step, through a series of transformations, we encode the input into a multidimensional embedding space. The manifold hypothesis states that real-world high-dimensional data lie on low-dimensional manifolds embedded in the high-dimensional space. Thus, you can imagine that each transformer via linear and nonlinear operations stretches and squashes manifolds in this space, passing the transformed manifold onto the next layer. We don't know if the manifold hypothesis is true, but it minimally provides us good intuition as to what's happening at each layer. The result of this step is that each input word is embedded in this space. Embeddings are ways for us to represent symbolic information numerically. Here, each word gets represented as a vector of floating point numbers, each of which represents some feature about the word. It's a way to convert high-dimensional data into a low-dimensional space. What's interesting here about this embedding space is that both distance and direction have meaning. Hence, sometimes you hear the term vector embeddings. From this embedding, we can learn abstract concepts not explicitly represented. For example, king is to queen as man is to woman represents the abstract concept of gender. Walking is to walk as swimming is to swam represents verb tense. In the second step, we decode the input sequence of words in the embedding space and eventually output the word with the highest probability of occurring next. But unlike DNNs of the past, critical to the success of the transformer architecture, are these attention layers, which I will explain shortly. In any case, we output bone, which is then appended to the previous output tokens and used as input for the next iteration. This is how ChatGPT works. Attention layers provide context for the words being processed. Context constrains the possibilities. Each node provides key information about itself to others. For example, I'm a noun. And each node can query for information from its neighbors. For example, I need a color. This context can help us resolve ambiguity in language. For example, Winograd's schema, which are considered mini Turing tests, are easily solvable by large language models, and thus makes it seem as if LLMs can do common sense reasoning. Here it's ambiguous whether it refers to trophy or suitcase. We would know how to resolve it if we know whether the last word is large or small, which might be in the context of a longer input sequence. Okay, let me take a step back from AI and talk about science and how it can benefit from AI. Generative AI can help scientists generate synthetic data, generate simulations, and more interestingly, generate new hypotheses. The novelty of these new hypotheses is that because a computer has access to an enormous amount of data, it can find patterns or correlations that would never occur to a human. AI can help us identify and discover. We can classify and predict objects, recognize and discover new patterns, and detect anomalies and rare events. AI can help us design and optimize experiments by helping us determine what control parameters to try for the next experiment, for example, on a large, expensive instrument. It could even propose new experiments for us to run. It's a given that AI can pore through scientific literature, summarizing results and creating compelling visualizations. All of these techniques are agnostic as to what field of science you work in, so it has the potential to expedite cross-disciplinary work. And there is a multitude of diverse sources of data to train and test new AI models for science. I'm now going to convey how AI has already been helping make new discoveries in astronomy and material science. Astronomers use DNNs to recognize galaxies and now can classify galaxies with an accuracy of 98%. Astronomers used AI to detect new exoplanets, predict signatures of new types of gravitational waves, and find a unique object that may be a remnant of two black holes merging. And astronomers use generative AI to produce a sharper image of the very first image of a black hole. In material science, the design space is huge. A short polymer with 100 amino acids has on the order of 10 to the 130 designs, more than the number of atoms in the universe. Material scientists are using generative AI to create new material designs. This is one example of a never-before-seen design of a hierarchical mycelium-based composite. Material scientists are exploring how to use AI to identify new equations and algorithms, to synthesize complex novel proteins that don't exist in nature, to visualize complex systems, and to predict how a new material will behave. Related to material design, our second speaker, Daphne Koller, will talk about the discovery of new therapeutic hypotheses in the context of drug design. Looking ahead, already the scientific community is exploring how to build foundational models for their scientific domain, which can be later fine-tuned to a specific problem. I'm actually curious to see how scientists will tailor concepts from the transformer architecture model. What are the analogies to predicting the next word, and to abstractions such as language, grammar, embeddings, and context? While I spoke about big data and big compute, for many reasons, including access to data and compute and enormous energy usage to build current AI models, the AI research community, especially in academia, is pursuing what can be done with small data and small compute. I'm very excited also to see the AI community interested in combining symbolic models of the past with statistical models of today. A different hybrid approach in science is to combine machine learning with physics-based models, for example, for simulations. Mike Pritchard, our third speaker, will talk about this hybrid approach for climate modeling. There are some challenges, the first of which is having enough data, which Jennifer Listgarten, our last speaker, will address. And finally, AI raises a new challenge to ensuring scientific integrity. To address this challenge, we need not just to educate scientists to check the accuracy of AI outputs, but also to do more research on trustworthy AI. Thank you very much. So now I'm going to introduce our first speaker, Pushmi Kohli. I have to just say that I knew Pushmi when we both worked at Microsoft, so it's such a pleasure to see him again. Pushmi leads AI for science, reliability, and security at Google DeepMind. His team at the London-based research lab has achieved landmark breakthroughs in scientific discovery using AI, such as 2021's AlphaFold. His talk today will explore how Google DeepMind is unlocking a new era of digital biology using AI. Pushmi?
Thank you, Jennifer, and it's a real sort of honor to be at this event to tell you about some of the work that our team is doing at Google DeepMind. So the topic of my talk is leveraging AI to advance science. I wanted to start by giving a description of our organization, Google DeepMind, which has the mission of building AI. Oh.
They have Daphne's talk up there.
I think we need to replicate this slide.
A synchronization problem here.
It's fine. I can use this one. Yeah. Yeah. So, as I said, our mission statement is to build AI responsibly to benefit humanity. And one of the best ways we see that we can benefit humanity is by advancing science, because science has the opportunity to tackle some of the most challenging problems that we are facing as a society. If you think about what is the need for AI, and if you look at scientific problems across the board, you see two sort of key trends. One is the scale of the experimental data that we are gathering about our world. From any discipline, whether it's biology, genomics, protein science, meteorology, we are collecting massive amounts of data about the systems that we want to study. At the same time, the complexity of the models that we have to deal with in experimental tools, like if you want to control, say, a tokamak, the sophistication of those tools is increasingly and becoming very sophisticated. And we need all the help that we can get from these machine learning and AI systems. The idea behind our strategy, our research philosophy at DeepMind on advancing science was to focus on root node problems, which we think can unlock the most pressing problems that are facing humanity. What do I mean by a root node problem? A problem like protein folding or quantum chemistry. If you somehow understand the structure of proteins, then that informs its function, and that can unlock progress in a number of different problems, like protein design, like understanding diseases, that has consequences for our drug discovery, enabling synthetic biology, and so on. In the same way, if you can somehow unlock quantum chemistry, then that gives you the ability to do material design much more effectively, opening up new avenues for designing better batteries, finding high-temperature superconductors, and so on. Our philosophy in approaching these problems was to not consider AI as a hammer looking for a nail, but to approach these as a true multidisciplinary Apollo program, where we bring the best from the scientific community that has been tackling that problem and merge it with data and the best machine learning and engineering principles that we have learned over many years. I'll try to explain this in the context of a number of case studies, with AlphaFold being one of the prominent ones. So what is protein structure prediction? So what is a protein? Proteins make all of us. We are essentially just bags of water without proteins. They are the building blocks of life. A protein is just a long chain of amino acids. It's represented as a string of an alphabet of 20 letters, which represent the amino acids, and they are the building blocks of life. All living organisms that we know of are constructed by proteins. They are the Lego blocks. This is how a protein looks like. It's a sequence of amino acids. Each 3-letter combination is an amino acid, and once you have this whole sequence, it constitutes a protein. The protein structure prediction problem is to, given the sequence, find the structure. And you can see these structures can be quite sophisticated. In this case, this is the structure of hemoglobin, and the hint is when the figure rotates, you can see the pocket where the oxygen binds. Now, what's the importance of protein structure? The structure informs function, and it has been a grand challenge in biology for the last 50 years. In fact, a member of the National Academy of Sciences, Christian Affinson, won the Nobel Prize in 1972 for sort of the hypothesis and his work on proteins, where he made the hypothesis that the structure should be determined by the amino acid sequence. Over many, many decades, the structural biology community has worked very hard on this problem. Just to find the structure of one single protein, sometimes it can take many, many years, like PhD worth of time to find the structure of one single protein. And the structural biology community has collected very diligently all these structures in a database called the PDB, which provides an amazing resource for training machine learning models. So this was very visionary of the community. Until now, you have 200,000 structures that are known in that database. But in the world that we know, there are billions of proteins. And so how do we expand, how do we scale up our knowledge of the living world and nature by finding structures of these proteins? That was the challenge for AlphaFold. Now, another key element that enabled the development of AlphaFold was the importance of clear metrics. The structural biology community had been had constructed this critical assessment, like the Olympics of protein folding, which was held every 2 years, which was done in a completely blind way. When new discoveries were made, they were not shared with the community, they were blocked in, and people were asked to make predictions about them. And so all the different techniques can be assessed independently in a completely sort of fair way. And that was another sort of key element. So both the availability of data and the presence of a clear metric were extremely important. If you see how progress on protein structure prediction was made through the critical assessment in the sort of decade preceding DeepMind's entry, the progress sort of somewhat had stagnated. There was incremental improvement over a decade. We started the AlphaFold project in 2016, and when we submitted it in 2018, we saw that we had not only achieved a state-of-the-art, but sort of done it so in a dramatic way. That gave us even more confidence to focus on the problem and, in fact, to really try to attempt to solve it completely. In order to do that, in fact, we had to put our original solution in the trash and start fresh, because we believed that we wanted to find a solution which is elegant enough that can go all the way. And our belief in machine learning actually showed prove results, where we revealed AlphaFold 2, which reached a level of performance which the experts felt had found a solution of the problem. Just to give you an indication of how far AlphaFold 2 was in its time, the next best entry in the protein competition had 3 times the error of AlphaFold 2 at that time. And here are some of the qualitative structures that AlphaFold 2 was producing. The ground truth is in green, the prediction is in blue, and you can see how closely those match. The structure that you are seeing on the extreme left is the structure of the ORF8 accessory protein of SARS-CoV-2, while the spike protein of SARS-CoV-2 was very quickly found out, the structure of it, but the accessory proteins, their structure was not known. And with AlphaFold 2, we made those predictions and revealed them with shared them with the scientific community at the start of the pandemic. Over the last few years, AlphaFold 2 has had massive impact. There have been 1.7 million users of the AlphaFold database, which contains the structures of AlphaFold that we have shared with the wider world. It was considered the breakthrough of the year by Science, method of the year by Nature. Like, one of the key surprising moments as a computer scientist when I was looking at it was 1.7 million, and now in fact it is 1.8 million people have used AlphaFold database. The fact that there are 1.8 million scientists working on protein structure prediction should be a note of extreme optimism about the state of the world and science. So we made these predictions, shared, and in partnership with the European Microbiology Lab, shared them with scientists all over the world, and they can use them in any sort of way possible. Now, and as we have seen, AlphaFold has led to accelerating progress on a number of downstream applications, from developing plastic-eating enzymes for controlling plastic pollution, blocking antibiotic resistance mechanisms, determining structure of the nuclear pore complex, a fundamental discovery in structural biology, accelerating drug discovery in neglected diseases, designing more effective malaria vaccine candidates, designing of new drug delivery mechanisms, and so forth. So there's a lot of impact that AlphaFold has achieved. But there was also a very humbling note when we released AlphaFold. After the release, we got emails from parents who were trying to treat a sick child, and there was nothing that AlphaFold 2 could do in that space. So it just shows you that while we had made amazing progress, there's a lot that needs to be done. And that's where we are going after AlphaFold 2. The next generation of AlphaFold goes beyond proteins and tries to find the structure of other biomolecules, RNA, DNA, small molecules, and how they interact with each other. We want to also go beyond proteins to general biology, understanding genes, cells, and organisms as a whole. So we are working on functional genomics. We are trying to go from sequence to molecular phenotypes. We are going from, in cell genomics, we are trying to understand the logic of the cell, going from molecular phenotypes to cell phenotypes, and this is very relevant for people studying cancer. And then in human genetics, we want to understand the disease. We want to go from sequence and cell phenotypes to organism phenotypes. Just to give you an example of some work in this space, we have done a lot of work on understanding our DNA, our genome. There are two parts of our DNA, the coding part, which code proteins, and the non-coding part, which control gene expression. Any mutation on the coding part can influence protein function, and these are called missense variants. And on the non-coding part, the mutations can affect influence of protein expression. We have done work on both. We published this paper last year on accurate proteome-wide missense variant prediction using our new model, alpha missense. And then we have also done work on applying machine learning for gene expression prediction. I'll give you a sort of short note on alpha missense. Only 2% of the observed missense variants are annotated in ClinVar, which is the key database repository for such kind of variants. Only 0.1% of known pathogenic of variants are known to be benign or pathogenic, and only 5.5% are seen in the human population but we don't know whether they are pathogenic or benign. With alpha missense, we could run over all the 71 million possible human missense variants and make predictions about whether they are likely benign or pathogenic, really expanding the work in this space. Our work is not limited to biology, and I'll try to give you a snapshot of other efforts that we are doing in this space. With our partners, EPFL, we are trying to use machine learning for doing plasma stabilization in tokamaks. A key problem in plasma stabilization is to develop control policies which can keep plasma stable in a tokamak. The difficulty of it is plasma is a very difficult thing to maintain a stability of, and there are many other issues like heat flux and so forth. What we did here was train a machine learning model to learn to control these things in simulation over millions and millions of episodes, and showed how it can transfer that experience into controlling a real tokamak. And with this, not only can it stabilize classical plasma configurations, but could control novel plasma configurations that scientists wanted to study, like elongated conditions and so forth. We are working on mathematics and computer science, where we are sort of going beyond the knowledge of can we discover new algorithms for fundamental operations like matrix multiplication, finding new connections in basic applied mathematics like in topology, where we have found a connection in algebraic invariants and geometric invariants in knot theory with our collaborators, Mark Andras, Jordi Williamson at ANU. And the progress is not stopping. It's in fact accelerating. Even in the last few months, we released 4 different AI models for 4 different things. The genome model, which discovers 2.2 million new crystals, including 380,000 new stable crystal materials for material discovery. Only 40,000 of these were known earlier. GraphCast is a model which can predict weather conditions up to 10 days in advance more accurately and faster than conventional methods. And instead of using a supercomputer, you can do that on a minute on one single chip. That just transforms who can use weather predictions and for what applications. I don't have a lot of time, but there are similar sort of advances that are being made in a number of different disciplines in our team. Finally, with LLMs, we are opening up a new era where not only can we use structured data, but we can now use unstructured data and knowledge that is embedded in all the publications in the scientific community. To summarize, I think AI is an incredible tool that can help us address extremely challenging problems that society is facing. And we are at the start of a new era of scientific progress with AI at the center. Thank you very much.
Our next panelist is Daphne Koller, who is CEO and founder of InCitro, a machine learning-enabled drug discovery company. She was a Stanford computer science professor when I knew her best, co-founder of Coursera, one of Time Magazine's 100 Influential People, and a MacArthur Fellow. She's a member of the National Academy of Sciences, the National Academy of Engineering, the American Academy of Arts and Sciences, and is a fellow of AAAI and ISCB. Daphne?
Thank you very much. It's a pleasure to be here. And thank you to Jeanette and Pushmit for so nicely setting up what I'm going to talk about. So last year was viewed by many people as the dawn of AI, but really what we've been on is an exponential curve. And the challenge of an exponential curve is that early in the curve, you don't really know you're on the exponential curve because the delta from last year to this year doesn't seem all that large. But as the curve continues to evolve, all of a sudden it's like, oh my God, I'm on an exponential curve. And I think last year was the year where everybody realized what several of us had known for many years. What led to this exponential curve is really the convergence of four forces. First and foremost, and Jeanette and Pushmit both alluded to this, is the unbelievably amounts of data that are now available to train the machine learning models. Those are accompanied by an equally incomprehensibly vast amount of compute power that allows us to interrogate those data, juxtaposed with an increasingly large number of people who can actually build these sophisticated models. And it is that has led to the machine learning models that are now so visible and so present in everyone's minds. So when you think about the problem that I'm going to talk about, which is can AI, in addition to all of the problems that we are familiar with, help us make better medicines for patients? The answer is we believe yes, but it requires all of those things that we talked about. We need the right data in the hands of the right people so they can build the right machine learning models. So this all starts with data. So the question is, how do you get the amounts of data in biology and specifically in drug discovery that can empower this type of AI models? So in our work, we've tried to build a large and growing corpus of human clinical data so that we can really talk about what might make a difference to patients. And juxtapose that with a data printing factory that can allow us to print cellular data at unprecedented scale. Those are integrated and interrogated using modern-day machine learning, and then we overlay on top of that human genetics, which is one of the very few sources of ground truth for causality that are available in a field that is rife with associations that are oftentimes not causal. So in order to enable this, we've created this infrastructure for collecting data in both humans and in cells. And because biology is so complex, it requires a multifaceted perspective in which multiple data modalities help provide convergent perspectives on what is going on within a biological system. And so we've done that in both human data and in cellular data to create this integrated perspective. Now, also importantly, as of today, we do not have enough data to really empower the kind of analyses that we need to do. And so what we've also done is brought into bear the power of generative AI to create additional forms of data that did not exist in the original. So what we're doing is imputing from limited data observations, other observations that were not made in a particular system to get larger and larger cohorts to power discovery. And I'll show you some examples of that in the work that we're doing. So on the human cohort data, we've focused on data that are available as part of the standard of care. These are data that are routinely collected, increasingly in digital form. They're oftentimes high content, oftentimes at multiple biological scales, and then we're able to use that to derive novel phenotypes, understand disease progression, as well as you'll see via the power of imputation, create relationships between data modalities that empower discovery. Similarly, on the cellular data, we've built this data printing factory with a predominant focus on single-cell data that is multimodal and high content, oftentimes far beyond human interpretation, and again, used AI to both increase the amount of data and create relationships between data modalities. So here's one example of a component of our data printing factory that we found to be incredibly powerful. It is a pooled format, high content genetic screening platform. So you have a population of cells. They can be uniform or they can be diverse. You introduce into those cells a pooled CRISPR library that edits individual cells, each one with a different genetic edit. So now you have a very diverse population of cells, each with its own genetic composition. We're using high-content microscopy, both fixed cells and live cell imaging, to phenotype these cells, each one on its own. So you have a very high-content phenotypic representation of each of those cells. By the way, none of that could be done without AI underneath the hood to empower these microscopes. And then having phenotyped each of those cells, you fix the cells, and you're able to read the barcode associated with each CRISPR guide so that you understand the genetic composition of each of those cells. So all of a sudden, you have in a single plate, 100 million experiments of genotype-phenotype relationships. And that is an incredible amount of data that can really power our understanding of the genotype-phenotype connections at the cellular level. So this is one example of the platform that we've built to robustly generate data at scale, derive machine learning-enabled insights, and then use that to make data-driven decisions on what are therapeutic hypotheses that we want to turn into drugs. So I'm going to show you some examples of this in practice, and so that you can see how one can leverage this. And we have efforts across metabolism, neuroscience, and oncology, and I'll show you a quick vignette in each of them. Metabolic disease is one of the nicest end-to-end examples of our work because it actually brings together both the clinical and the cellular data into a single integrated approach. So what we do here is we start with human cohort data, which are experiments of nature that give us genetic diversity aligned with human clinical phenotypes. We use that alongside with statistical genetics to derive novel insights about genes and pathways that are implicated in disease. And then we use that to define the experiments that we perform in this automated lab that I showed you a moment ago. And those in turn are interrogated back in the human data in order to give rise to truly high confidence hypotheses that we take into drug discovery. So in this work, we set out to interrogate the genetics of liver disease, fatty liver disease, as one example. And here it's a case where fatty liver disease is highly underdiagnosed because it requires very expensive and often very invasive testing in order to ascertain disease in a human. And as such, there's very few cases in which the disease has been ascertained, and the power for genetic discovery is quite limited. We leverage the power of machine learning to take data modalities that are much more abundant, much more readily available as part of the standard of care, and we're surprisingly able, via machine learning, via AI, to impute quantitative liver fat for modalities where most people will have told you it is impossible to see liver fat in DEXA scans, which are a bone scan, from circulating blood metabolites, or from 37 circulating blood biomarkers. Now, this is not perfect predictive accuracy. DEXA is actually pretty good. The other ones are more modest predictive accuracy, but are far better than a human could get and allow us to take and impute these quantitative liver fat measurements in much larger scale than you would ever be able to get via clinical data. And what does that do for us? If we then do genetic discovery associating genes in the genome, so here the x-axis is the genes on a chromosome, the y-axis is the strength of an association with the clinical phenotype, which is liver fat. If you just use MRI as a modality, very few patients, very few discoveries that pass statistical significance. If you start going into these additional modalities, which admittedly are somewhat lower quality but are much better powered, all of a sudden you see a much greater number of statistical findings emerging, including, for example, the first genetic support for a target that we know works in the clinic, FGF21, as well as a range of multiple novel genetic hits. We can now take that and interrogate that in the context of what it tells us about the pathophysiology of the disease. So, for example, if you look at the original genetics, there's, again, very few genes that have been associated. Very little insight emerges on what are the pathways that drive the disease in the liver. When you take the new, much richer genetic architecture, de novo lipogenesis, which is the generation of lipid droplets in the liver from its substrates, emerges as by far the most significant pathway, which is a novel inside. We can take that and create now a cellular system to put into these high throughput data generation that instead of using free fatty acid as the condition, uses the de novo lipogenesis substrate. So an AI-informed experimental design that allows us to now screen in the system and identify new genes that potentially invert the de novo lipogenesis phenotype. So what you see here is on the left side, lots of these little green droplets, which are lipid droplets. In the middle is a positive control, in the right is a positive control. In the middle is one of the genes that came off our screen, and you can see a significant amelioration of these lipid droplet phenotypes. When we took that, aligned it with the human genetics, identified a small handful of high confidence targets, and towards the end of last year put them in an animal model, an OB mouse model, and were able to show a considerable knockdown of the triglycerides as well as other phenotypes, including cholesterol in circulating bloodstream, that really justified these findings that we had. Okay. Very quickly, this was one example. In neuroscience, we are leveraging the same thing, focusing primarily at this point on our cellular system. The disease in question here is ALS, which is, as we know, a devastating disease with effectively no disease-modifying treatments. We were able to take induced pluripotent stem cell to turn them into motor neurons at unprecedented scale. And it turns out that in these images, machine learning can see things that a person will never be able to see. For example, just by looking at cell morphology, the machine can predict the mislocalization and aggregation of TDP43, which is the hallmark of this disease in humans. So you can see a very strong association between the measured and the imputed TDP43. You can also, by looking at these cells, impute changes in expression of individual ALS-related genes, again, at fairly high accuracy. And that allows us to use this optical screening system that I talked about earlier to now screen for modifiers that revert these ALS phenotypes into something that looks more like healthy cells. And we've been able to identify a handful of targets that we're now pushing into drug discovery in this system. In service of this effort, we've established one of the largest collections of ALS genetic lines, corresponding both to engineered isogenic strains where you have a healthy cell and one mutation, so you can do like an in vitro case control study, familial lines as well as sporadic ALS lines. We have the ability to take those lines and turn them into motor neurons in 10 days, all in a completely automated, hands-free way, so you get very high-quality lines, and then use this multimodal analysis to understand the phenotypic landscape of the genetic diversity of ALS and understand how different genetic backgrounds, all of which ultimately give rise to the death of motor neurons, what are the trajectories that those cells take, and therefore what are some potential interventions that we can screen for that might stop those trajectories early before they result in the death of the motor neurons. Finally, to give you yet one more brief vignette as an example how we're using this in the context of oncology, here the focus that we've started with is actually starting on the clinical side. We've focused on a data modality that we know is very abundant and incredibly information-rich, which is the standard histopathology slide that every single solid tumor patient pretty much anywhere in the world gets as a biopsy when they come in to have their for a diagnostic procedure. So this is a very abundant modality, increasingly in digital form, has incredible information content at multiple biological scales, ranging from organ-level things like vasculature, through cell-to-cell interaction, all the way down to intracellular structure. You give this to a human pathologist, they give you 3 ordinal numbers as the information extracted. AI can extract incredible amounts of information from this. So as an example, we start out by taking these histopathology images. We apply the kind of self-supervised language learning models to construct an embedding in a self-supervised way. And then we can overlay that with both clinical data on the one hand to understand how different featurizations, different features of those histopathology slides give rise to differentiated patient outcome. And on the other hand, you can now overlay molecular data on top of those same samples to understand what's going on underneath the hood from a molecular basis within those images. And by putting those together, you end up with novel therapeutic hypotheses. So let me give you one example. This is taking those embedding images from those patients, and you can, by using those embeddings, predict with very high accuracy DNA alterations that we know are drivers of cancer. This is what you see on the left in CRC and ovarian cancer. This is true in other cancers as well. On the right, you can see the ability to predict subtypes of the cancer that are defined typically via measuring RNA activity within those samples. Turns out you don't need to measure the RNA. The histopathology on its own gives you very high accuracy predictions of those RNA-defined phenotypes. And that turns out to be not an accident. What you see here is that, in fact, you can impute from those H&E samples both ATAC-seq, which is basically measures of open and closed chromatin, open and closed DNA. You can predict that from the H&E, not for every peak, but for a large subset of peaks. And RNA expression, you can predict for fairly large fractions of the transcriptome with very high accuracy. You can see the R-squared is 0.64. On the right-hand side, you see one example of predicting for a set of therapeutically relevant targets on a per-patient basis which patient has that target expressed or not expressed. So the beginnings of molecular precision oncology, by looking at those imaging samples alone, you can predict, is this target expressed, and therefore, presumably, whether that is going to be a therapeutically relevant intervention for that particular patient. So we're using this to both drive the opportunity for precision interventions within patients, as well as to engineer therapeutics that are likely to be relevant for particular patient populations based on what we can see in this imputed molecular data. So to wrap up, looking ahead, I'm going to turn to a quote by Eric Schmidt, which to me is, I think, really captures it well, which is, "Math is to physics what AI will be to biology." You needed math to understand how physics work. You'll need AI to understand how biology works because it's so incredibly complicated. So with that, thank you very much.
Great. From biology to drugs to climate. Our next speaker is Mike Pritchard. He is 50% faculty at UC Irvine and 50% research director at NVIDIA. At the university, he has explored using machine learning for hybrid climate simulations to bypass the computational expense of high-resolution physics. While in industry, he's exploring whole atmosphere machine learning emulation and AI for downscaling weather and climate extremes as part of NVIDIA's Earth 2 initiative. Mike?
Thanks. What an honor to be among such esteemed company. Thank you for having me. So I want to start with weather. I'm a climate scientist by training, but I've had a very disruptive 2 years working with professional deep learning engineers, and they've opened my eyes to whole atmosphere weather simulation. So weather simulation used to be about physics equations, solving F equals ma for an incompressible fluid on a rotating sphere with big supercomputers, plus a lot of tricks to deal with the stuff that we can't actually solve computationally, like clouds and turbulence. Nowadays, data-driven weather prediction feels a lot more like making this woolly mammoth image. How many people have seen Sora videos on the internet? Look them up. OpenAI's video generation with GenAI is amazing. But it's basically like taking machine learning and asking it to do video prediction, but instead of having 3 channels to deal with, red, green, and blue in a pixel, you've got lots more channels, temperatures, winds, and pressures at different altitudes of the atmosphere. And instead of having the whole internet to train off, which makes these stunning woolly mammoth videos, we have 15,000 days of satellite records. But nonetheless, this is producing skill gains rapidly. 23 was a breakthrough year. We saw earlier, DeepMind took the cover of Science with their GraphCast model, which is a nested icosahedral hierarchical GraphNet. And the European Center for Medium-Range Weather Forecasting, which is the world's premier physics-based weather forecasting agency, has started building their own AI weather model as well. It's not very controversial how to score weather, and so here's a scorecard hosted by Google Research. The main point here is there's a lot of blue boxes. Every box is a place where a particular AI weather model is predicting a particular variable at a particular lead time between 1 and 10 days. And where they're blue, they're beating the deterministic physics benchmark, and there's a lot of blue boxes. The European Center for Weather Prediction is hosting these AI forecasts, so it's such that more and more meteorologists are looking at the details under the hood of these aggregate skill scores and finding them convincing representations of patterns. This is something that's been very convincing to me. Once trained, despite the fact that they've been trained on the noisy soup of real weather, these models can be interrogated for whether they've understood physics. And so the act of predicting frame-ahead weather surprisingly learns a more generalized dynamical operator than I might have thought. And proof of that is that you can take the model after it's been trained and expose it to tests, idealized tests like this, where there's a bullseye of pressure on the equator. And what we see is a classic geostrophic adjustment where convergent flow eventually turns into rotational flow, especially off the equator. And these sorts of canonical idealized physics emerge in these AI weather models despite the noisiness of their training data. And there's a lovely paper that ticks through a bunch of them if you're curious. But the main breakthrough is the fact that once trained, these models are very fast to inference. They're thousands of times faster than doing the true physics calculations, such that for all of history, there's been this tension in weather simulation where you'd like to have more resolution because some impactful phenomena like hurricanes require high resolution to simulate faithfully. You'd also like to have more ensemble members because weather's a probabilistic prediction and we'd like to resolve the histogram of possibilities and especially the tail so we can hope for the best but plan for the worst. But there's always been a tension here under a limited compute budget, and most weather forecasts are on the order of 50-member ensembles. But once for a given resolution, inference becomes trivial, that's game-changing. And we can start to look at low likelihood, high impact extremes. So this is a very important topic in modern, at the interface between climate and weather. For instance, there's this Pacific Northwest heat event that ignited a small town in Alberta a few years ago. It's very difficult to study the climate drivers of low likelihood, high impact extremes like this because there's so few in the observed record. So the only alternatives are theory or simulations of plausible observations. And AI weather models give you a lot of simulations of plausible observations. So I think this is one of the most interesting scientific use cases that's emerging for this technology. I'd like to credit Bill Collins and Ankur Mahesh at UC Berkeley and the Cascade team, which has been studying climate drivers of extremes for decades. They've recognized that you can do some retroactive counterfactuals of historical climate with this technology. So they've taken a fit-for-purpose calibrated version of NVIDIA's weather model called ForecastNet and tried to reforecast this Pacific Northwest heat event. In the gray lines, you can see the 50 members of a conventional ensemble, which none of them 10 days in advance are able to predict the red line, the observed Six Sigma heat event. But 1 out of 7,500 members of this huge AI ensemble was able to capture such an extreme event. And this is what it looks like, that one ensemble member. It looks like atmospheric physics, Rossby waves that happen to have conspired under this particular manifestation of internal variability to create a large persistent ridge, pressure ridge over the western US, consistent with a realistically large and intense heat event. So that's just evidence emerging from the new field of huge ensemble science that's being powered by AI weather. Now, so far I've only discussed the medium-range timescale, 1 to 10 or 15 days. The atmosphere loses its memory of its initial conditions on that timescale. But there's other sources of predictability on longer timescales involving the ocean. I forgot to point out this. Let me divert for a moment. Such huge ensembles only work if you're meticulous about calibrating a weather ensemble, and the art of probabilistic calibration is non-trivial. But I wanted to point out a few studies this year that have made good progress. The first is from Google Research Neural GCM. The equation there I've demonstrated emphasizes some terms they've added into the AI's optimizer that make it aware of the fact that different variables in the atmosphere decorrelate at different timescales and on different wavelengths. And allowing the optimizer to be aware of this is useful for it to discriminate signal from noise. and also adding more than one ensemble member inside the optimizer and including a term that favors realistic trajectory divergence. Together, these sorts of loss manipulations can produce reasonable ensemble probabilistic calibration. The Hens paper, I won't mention how they've achieved good calibration, but there's a paper brewing you'll be able to read in a couple months. But I did want to emphasize DeepMind's latest contribution to global weather forecasting, which is formally probabilistic. It's a method called GenCast that uses diffusion models to be probabilistic about the problem from the get-go. And I think there's some really interesting results there worth emphasizing. So bear with me, but everywhere there's a blue line underneath the black horizontal line here is a place where this probabilistic AI weather model is beating the physics gold standard, not on a deterministic skill metric, but on a range of probabilistic metrics. So on the left, the root mean squared error of the ensemble mean. In the middle, the continuous rank probability score, which penalizes both overconfidence and the ability to include the observations in a PDF of predictions. And on the right, I think especially impressive, a Breyer score, so a discrete probability for exceedance of an extreme climatological threshold, 98th percentile wind values. So the fact that we're seeing 10 to 15% better than ever skill emerging for such probabilistic forecasts, I think, is the latest milestone in AI disrupting weather, and that's just emerged in the last half year. Remains to be verified by meteorologists. Okay. But I mentioned the transition from weather to climate and the loss of initial conditions after a couple of weeks. Seasonal forecasting requires months, and climate forecasting requires decades of simulation. This is a milestone that Dale Duran at the University of Washington revealed at NVIDIA's GTC conference last month, showing a 100-year rollout from an autoregressive machine learning model that includes ocean coupling. And in the bottom graph, it also includes information from satellites about long-wave radiation that captures high cloud feedbacks critical to El Nino. And you're looking at a time series of sea surface temperature in the Central Pacific, which is where El Nino lives. And indeed, a few years into the simulation on the bottom compared to the top, the observations, the pattern of El Nino seems to be emerging in ocean-coupled AI autoregressive forecasts for the first time. So there's potential on seasonal timescales here. Okay, so that's weather. Now, I'm interested in climate, and I've been tinkering with trying to infuse AI into climate models for a few years, so I'd like to talk about that. The first thing you can do is take this AI weather technology and apply it to future climate simulations. There's troves of output from coarse resolution climate models, and that's starting to work. This is a team at the Allen Institute who took NVIDIA's weather model and trained it on climate model output, and they're able to reproduce 10 self-similar seasonal cycles, which is a necessary condition to replace the atmospheric component of a climate model with an AI equivalent. You have to do that across many components of the Earth system for climate simulation. These are examples from New York University showing on the left a deterministic ocean prediction with vacillations in the Gulf Stream temperature, and on the right-hand side, 1,000 days into an ocean emulator producing similar dynamics. But I think really climate simulation is a subgrid parameterization nightmare. Climate simulations have to simulate the whole world for hundreds of years, hundreds of times to beat down internal variability across dozens of what-if emission scenarios. And you just can't afford much resolution when that's the problem. And so all of these processes are heavily approximated in climate models. And a really ripe frontier of AI for climate simulation is being more ambitious in our representation of those processes by training individual emulators of critical processes like radiative transfer, like cloud microphysics, like oceanic mesoscale eddies, and aerosol chemistry. And we often don't represent these processes with the fidelity that we think they deserve just due to compute constraints. But for the same reason that AI weather simulations are so much faster than conventional weather systems, that changes. And so these are places where individual processes are already exploiting AI for parameterization. Now, in my opinion, the holy grail is multi-process subgrid AI emulation because that's where you get the most computational benefit, and climate is such a compute-limited problem. There are tantalizing glimmers of success on this front. This is one of my favorites from MIT a few years ago, Yana Yuval. On the left, imagine running a very high-resolution atmosphere model. It's slow to run because it's high resolution, but you can coarse grain it horizontally and learn carefully the subgrid flux corrections needed for coarser scales. And if you're successful, then run a coarse resolution model that's much faster to integrate, but is constantly being corrected by machine learning subunits that are correcting it for all subgrid processes. And at least in this one case, in this idealized limit, that yields a simulation that runs hundreds of times faster but produces the same statistics. And so that's under the broader goal of using AI to make today's low-resolution climate models behave as if they were high-resolution, to do more justice to cloud feedback, to include more physics than is possible today. And there are at least 4 national centers blooming that I'd encourage you to follow if you're interested in that. The Climate Modeling Alliance at Caltech, I mentioned the Allen Institute, Professor Laura Zana at New York University and the Courant Institute, who's working with the Geophysical Fluid Dynamics Laboratory at Princeton and government climate modelers to build ocean emulators. And a center I'm affiliated with at Columbia, led by Pierre Gentine, called Learning the Earth with Atmospheric Physics, and NSF Science and Technology Center. But each of these centers are innovating new machine learning climate parameterizations every month, it seems. My own personal pet test bed is this sort of a simulation framework. It's called a multi-scale climate model that makes the idealization of representing many processes like convection and turbulence and radiation locally in an idealized two-dimensional subdomain. That's convenient because it eliminates the non-locality of the parameterization problem and allows us to focus on other challenges, such as multi-process emulation. You can think of it like this. On the left-hand side, picking a column of atmosphere and taking the macroscale thermal structure and thermodynamic structure, as well as relevant boundary fluxes of sunlight and evaporation, and then trying to replace the actual solution of a complicated cloud resolving model with machine learning to predict the heating tendencies and how convection vertically redistributes heat and moisture while making clouds that interact with radiation. And we've had a few successes and challenges over the past 6 years working on that problem. On the left-hand side, learning how to enforce mass and energy conservation that's vital to climate simulation turns out to be solvable. Improving generalizability to allow a spatially local parameterization, which you might have more hope of generalizing. We've made progress by making physical renormalizations that turn the extrapolation problem into more of an interpolation problem to achieve more climate invariant machine learning. Respecting stochasticity, of course, these subgrid processes involve clouds and turbulence. They are inherently stochastic, so one has to use modern stochastic machine learning methods. But there's ongoing challenges, especially as regards to controlling errors. So hybrid AI physics models are in their infancy, and they're difficult to control, and we're still learning how to control them and avoid them going haywire on the downstream task. And so lately, we've been trying to enlist the help of the machine learning community, and the way to scale that help is to build benchmarks that speak to them in the language they prefer with very clean data pipes. And so I'm proud that this consortium of climate scientists over the last year and a half managed to do that for the first time and got an outstanding paper at NeurIPS that will hopefully galvanize enough attention to machine learners to help us make a phase change in our ability to do hybrid machine learning for climate. So with that, I'll close with a couple of remarks. I hope you're convinced that AI is already here for weather prediction. There's unequivocal skill for deterministic prediction and encouraging progress for probabilistic calibration. And these calibrated AI ensembles are opening the door to new huge ensemble science that I expect to update our understanding of the climate drivers of low likelihood, high impact extremes and lead to better risk calibration of the current risk that we face in current climate, which is needed to calibrate our future exposure to future climate. It's exciting. The field is moving fast. Long rollouts have achieved just in the last year that include ocean coupling and the beginnings of El Nino dynamics. And I think it's fascinating, the evidence of learned physics that may justify more general use cases than weather prediction alone. For climate, full model emulation of existing coarse resolution climate models appears to be viable. How to couple those models together is still an unsolved question, but it's possible we'll be moving in the direction like ChatGPT drew for me here, where climate models will have more and more AI subcomponents. There's been definite success in emulating individual processes for radiation, microphysics, and oceanic mixing in chemistry, but challenges in emulating all of those processes together that have led us to create large new benchmarks. And I limited the scope of this talk to avoid the final 2 bullet points, but I want to mention their existence. There are equally important things going on in AI for data assimilation and AI for downscaling, creating high-resolution, infrastructure-relevant predictions from coarse-resolution climate predictions. So thanks for your time.
Thank you, Mike. You know, I knew before this panel that I had a connection with 3 out of the 4 panelists, but now I know my connection to Mike, which is you're part of the Science and Technology Center at Columbia on AI and climate. So good to have a connection with you. Our last speaker is Jennifer Listgarten, who is a professor at UC Berkeley in the Electrical Engineering and Computer Science Department and a member of the Berkeley AI Research Group, the Center for Computational Biology, and the Bioengineering Program. She joined UC Berkeley in 2018 after spending a decade at Microsoft Research, which is, of course, where we knew each other. Jennifer?
Can I use this mic? Yeah, okay, fantastic. Small people get stuck behind podiums. I might stand here. So, great. So hopefully a bit later I'll explain this picture is a little bit crazy. Oh yeah, I can see them there as well, right. So, okay, great, let me actually go back. So what am I going to tell you about? So I've been working at the intersection of machine learning, what we now call AI, and the sciences, in particular biology, for several decades. And now that in the past few years, ChatGPT has become very visible to many people and many of these other tools, a number of my colleagues and collaborators and more broadly have been asking me these questions about what is going to happen in the sciences with respect to AI. And then I got these questions kind of over and over. And so eventually, in frustration, I wrote this piece basically to answer it once and for all, at least in that static moment in time. And so I'm going to take you through basically that commentary right now. And I'm going to seed it with 3 particular questions that I got over and over. And then I'll address these after. So the first one was, since ChatGPT works so well, are we on the cusp of solving science with large language models? And so, well, why would someone say that? You've, of course, heard that GPT-4 can now pass the bar. It can do well on the MCAT. And so I found this recent paper on, these are actually graduate-level biomedical written exams. They're primarily short answer, which is half a page, quarter page. There's long answer. There's questions about figures in there. And actually, on all but 2, GPT-4 did at least as well as the average student. And on, I think, 3 or 4 of them, it actually did better than any single student. You'll see one where it failed, which is advanced genetics. Geneticists don't get too excited. I think that's probably from the lack of training data, which will be one of the themes of my talk. I don't know, that's a speculation here. But of course, this database was not geared to any of these topics. This is just whatever GPT-4 could do at the time. Okay, the second one, isn't the huge success of AlphaFold suggestive that the potential of AI in science is limitless? And unfortunately, some of the previous speakers have already talked about this problem of going from the sequence to the 3D coordinates of the protein, which give you the structure. And so and truly this was a phenomenal success. This was, I would argue, the only phase transition in success we have seen that AI unlocked in the sciences. And so in 2020, after decades of work on this extremely important problem, which has received several Nobel Prizes over the years without any machine learning, suddenly we got to, like, as Pushmeet said, like atomic level accuracy. So, and because of this, people started to say, okay, well, are we going to see this huge phase transition of successes in all kinds of important problems in the sciences? And now the third one, which has to do with the first image on my slide, is so I often say to people, we don't have that much data in many of the sciences. And they say, yes, but can we use AI to generate data to overcome the lack of data? Okay, so it's fantastic that you're laughing at that, because that's, I think, the appropriate response. But shockingly, I don't often get a laugh. I get this question very seriously. So actually, fantastic audience. I wasn't expecting that. Maybe I can stop talking, because it's kind of half the point of my talk. So why do people think this? Well, we hear about generative AI, and so we know ChatGPT. It's a generative AI. It generates text, it generates sentences, it generates creative poems, and we have analogs for this. We can generate proteins with generative models. We can generate molecules. And so genuinely, people ask this question, can we use AI to bridge the data gap? And Daphne mentioned a certain element of this, and it is actually quite a nuanced technical discussion, but I'm going to try to just at least dispel one common notion as I continue to talk. So now I've planted you with these 3 questions, and so now I'm going to just kind of pop up a level and get into these topics a little bit. So how is it that we get an AI win on a problem? Like, what is it that enables us to do that? And so this is a little bit, I'm going to paint this in broad strokes here, but one is we have lots and lots and lots and lots of data. And the other one is we have just the right problem. And of course, it's some combination of these 2 things, in fact. So now let's dig into each of these 2 things. So lots and lots of data. Who thinks they know what that means? Okay, these are the really visible tools that many of you probably have used at least one of these. These are all the text and image tools that have just been wildly successful, that are literally changing the world. So they do have this, they're fantastic, and yet, if you've followed the news in the past month, you've probably seen articles like this, which basically say all these companies are freaking out because they've run out of internet data. The internet is too small. Okay, so if they're in trouble, what kind of trouble are we in for the sciences? So let's get a sense of how much data is out there. So we don't know exactly what those companies are using, but a huge component of what they're using, and they're probably not changing anything by any orders of magnitude, is a database called Common Crawl. And the most recent estimate of how many web pages Common Crawl contains is 300 billion, which is stored as 400 terabytes. And every month, they get 3 to 5 billion new pages added. And still, these companies are freaking out because this is not enough data. Okay, so how much data do you think we have in the sciences? This is not comprehensive. These are just some examples of popular areas where people are currently working in machine learning. This is the basic sciences. I'm not talking about medicine or other things like this. So, okay, so let's take a look at these. And keep in mind, of course, this is not the same currency because the first line says number of web pages. The second one is number of protein sequences. These are different entities, but I think it still tells you an important story here. So already, if I go to the largest thing here next to web pages, I see the number of protein sequences. That's 300 million. ChemSpider contains 100 million chemical structures. And the next one, this is the key to AlphaFold's success, is there are 200,000, just 200,000 examples where you have a sequence and a structure. And this is sort of the, if not for that, AlphaFold would not exist. There's a number of places where people are trying to accelerate physics simulation by training machine learning models to reproduce them. Here's one example of that, which is Open Catalyst. It has 60,000 so-called relaxations for some oxides. And then the last one is actually something people are actively working on, predicting RNA structure. We have only 2,000 examples of those at the moment. Okay, so I originally was going to put it as a graph because I hate looking at numbers, but this is what the graph looks like. And in fact, I actually cheated. This is data manipulation. Those blue splotches don't exist, but I thought I'd put them there. So I think you get a sense of what's happening here. Okay, so, but size isn't everything. Aside from the fact that the currencies weren't the same, these were protein sequences, web pages, even if I put them in the currency of bits and bytes, it's still not, size is not everything. And so, for example, if we were to sequence every human being in the world, there's around 8 billion humans right now, that would be about, if I've done the math correctly, 1.5 gigabytes. Naively stored on a hard drive. But the thing here is that what actually matters is information content, right? And so because all of us share most of our genomes, it means that many of those letters are actually redundant, and you could actually compress that to be much, much smaller. And for many data sources, for all data sources, in fact, this is more or less true. And so even if you got it all down to bits and bytes, you'd have to ask, what's the actual information content there? And the other thing I've glossed over a little bit is that if those data are labeled, this is like pure gold kind of data. And unlabeled data are an entirely different story. And so the reason that AlphaFold could get away, in essence, with just 200,000 examples is that they were this precious, precious data resource where you had a sequence paired with a structure. And so, you know, the web is a bit complicated. You could say implicitly it contains question, answer in there, and so it's maybe somewhere in between. Okay, so now I've talked about what it means to have lots and lots of data and kind of where we are. So what do I mean by what's just the right problem? So if we think about all problems in the world, all scientific problems, then there's going to be some set of them that we could frame as a machine learning problem. And of those, there's some subset of them where if we framed it as a machine learning problem, we could evaluate how well a model we had trained is likely to do when we deploy it in the greater world. So an example is I could hide part of my protein structure database from myself, build a model, and see how well it does on the part that I hid, assuming that it's sufficiently different, like it's not cheating. And if I do that, I get a pretty good estimate. But if I'm actually designing a new protein, I cannot check my model in that same way, and that becomes a much, much harder problem. Okay, and now I guess you're expecting this one is, are there sufficient training data that exist or could be readily created in the near future? And so if you have all of these, it's probably a good problem to tackle. And so I would say that not only was DeepMind brilliant in its creating a very nice model for protein structure prediction, they did incredible software engineering, but I would argue they picked just the right problem in the sciences as well, and that's why we saw this massive, massive success. Right, so they had a very clearly defined problem that could easily be posed as a machine learning problem, which is I give you the sequence, you tell me the 3D coordinates of every atom in the protein. This can be very easily checked. I can hold some of these proteins out. I can run the prediction. I can see how far away the atom coordinates are. And finally, and this is really key, is that those 200,000 sequences, those were in the public domain. And I have a citation here. The conservative estimate replacement cost of those 200,000 data points is $20 billion. Okay, so like you want to start a new problem? Think about that number. And although it is a fantastic success, what often gets overlooked is there remains a very big open problem that's highly relevant to therapeutics, to protein engineering, which is we know nothing about how those proteins move, and that is a key thing. And why don't we know it? We don't have the data to do the machine learning. So it remains an open problem. But I don't want to downplay the fact that this is a fantastic success. It has truly changed the world in protein science. So that was just the right problem. But I think in the sciences now, people are sometimes trying to find which problems can we hit with machine learning. But we should remember that science is about curiosity. And we don't even know what some of the most impactful and interesting questions will be asked. And once we do ask them, they may not be suitable for machine learning. And if they are suitable for machine learning, the data may not exist. And we may not be in a position to readily generate those data right now. So that brings us back to this problem of lots and lots of data. And like I feel like in this audience, since you all laughed in the beginning, I could skip over this part. But so this is the part where genuinely a lot of people say, can we use AI to bridge this gap in the sciences? And so this is my attempt. It's a bit creepier than I wanted it to look. But this is like a generative model, which is generating its own data and eating it in a supposedly virtuous cycle. Of course, it's not a virtuous cycle unless you're very, very careful. Okay, so this is one of my favorite quotes because generative AI seems very magical at the moment. Phil Isola and colleagues have been presenting this idea, which I think is a fantastic description, which is the following. Generative models can be viewed as a compressed and organized copy of a data set. And like really, that's all they are. There's no magic. They're very useful. We can extract representations from them. They can help us to search in very high-dimensional spaces for the things we want. So they are fantastic, but they're not going to help us to bridge this data gap. So fundamentally, new information has to be injected somewhere into there. So as you all know, because you laughed, I cannot take a generative model, generate data, and then train that model again with the new data, right? This is a perpetual motion machine. But I could generate data from one model, and I could use it with a different model. Basically, as long as I'm combining new data sources, there is a win to be had. And generative models can provide a very beautiful and compelling way to combine data sources. And so I like to always quote my very good friend and fantastic colleague, Alyosha Ifros. He's a computer vision researcher, extremely well known for his work in generative models. And he says, a few times each year, a physician, and this is not a joke, a physician with fewer than 2,000 MRI images contacts me about using our generative models to generate more training data. Now I know which doctors not to go to. Thank you, Alyosha. Okay, so I guess my point here is that the only way forward for AI in science is ultimately to go back to the wet lab and to keep generating data. And actually, as I think Daphne alluded to, often as we're doing this, AI is playing an extraordinarily huge role, either behind maybe the imaging components, but actually we can use it to guide which data we want to measure so that we collect things in a more effective manner that's most likely to improve the machine learning model. So even in this particular scenario, there's a huge opportunity for machine learning, which is currently going on as we speak. So in closing, I just want to say that the raw ingredients and outputs of science are not found in abundance on the internet, where a lot of other data that's making things happen is. Generative models are fantastic, but they cannot bridge this data gap. And I actually personally don't believe that major open scientific questions are going to go through these phase transitions of success the way AlphaFold did with machine learning alone. However, I work at this intersection. I'm an optimist. I think we're going to continue to advance scientific progress at a rate not possible without machine learning. And I do think there will be some phase transition that happens. I don't know when. I don't know what it will be in, but I look forward to finding out. Thank you.
That was a fantastic closer, Jennifer. Good to get a laugh out of the audience. So we now take questions from all of you. There are mics. If you have a question, please go up to the mic and you can ask questions of any of the panelists. Please don't be shy. I don't think we have mics up here, so you'll probably have to come up here to... Oh, you have your own mic? Yeah. Okay, great. Okay, yes. Please state your name, perhaps an affiliation.
Hi, I'm Tim Springer. And for a novice, after reading Jennifer Liscard's paper, what is the best way for me to learn how to use ChatGPT-4 to solve my biological problems, or just help me be more efficient as a mentor and doing some of the things I do?
Jennifer, you want to answer that?
The question was, how could you use ChatGPT to effectively help?
No. How do I learn? How do I just go about the first baby steps and then progress from there?
For science or for anything?
Well, for science,
Okay.
and for a biologist.
Okay, Jennifer?
I didn't completely catch the question. It's how do you...
Maybe it was too naive.
How does he get started?
How do you get started to use GPT, or how do you get started with AI to solve your scientific problem?
Well, I was going to use ChatGPT,
Okay, I mean, I
but
think
maybe that's a mistake.
I see. I mean, to me, ChatGPT is like a brilliant, version of Wikipedia, the whole World Wide Web. Like, I use it all the time, and then I fact-check it by doing regular searches and seeing if the citation exists, and so forth. But I do think it's a fantastic literature search tool. And people are trying to use it to generate scientific hypotheses. And in fact, sometimes when I have a new idea, like scientific methodological idea, I actually give it to GPT, because if it seems to know the answer, then I feel like I don't have a super original idea. I mean, I go look to see what it is, but so I guess I just think it's a really fantastic interface to browse literature, and then you got to fact check it.
Okay, and then where do I go after that?
What do you want to do?
Well, you
next?
say to really
I would
use machine
suggest
learning.
Either perhaps another panelist take answer, or we go to the next question, and you two can talk offline. Daphne or Pushmeet, do you want to say anything about this?
I don't know. I mean, as Jennifer said, these are incredible sort of tools. Large language models, they have a lot of interesting information embedded in them, and they can be useful to sometimes sort of discover and do literature survey. They do hallucinate in their current sort of versions, but we will see sort of progress on that. One of the examples that I have seen is that you could use these models for really data processing in a way that is remarkable. You could, in one of the examples I didn't get time to show, there was a paper in genomics where an authors had basically carefully sort of gone through almost thousands of papers to understand what are the genotype-phenotype associations in a specific sort of domain. And they had done that till 2020. And there were many other papers beyond that in the last sort of 3-4 years that have been published. What the latest version of Gemini, which is another sort of LLM, was able to do, it was able to search for papers in that topic on the web, read all of those, process which of them talked about this specific genotype-phenotype sort of association, and augment the data in real time. And then when we did the validation as to how many of them were correct, they were mostly sort of correct. So this can really accelerate the productivity of doing literature surveys or sort of extracting information which is embedded in publications that frankly is growing at a rate that all of us are hard to keep track of.
So maybe if I can...
Yeah, that's very helpful. Thank you.
Maybe if I can just jump in, you should think of this as an army of really smart but not very well educated undergraduate students. So the kind of tasks that you would assign to them, you could assign to the GPT, and I think the 3 categories that I've found most useful are, first of all, this literature survey of a very large mass of literature. You've heard that. I think talk to your PDF is one thing that you could do. You could upload 1 or 2 or 3 papers and then actually interrogate sort of in a conversational way and ask questions that get to what you're looking for in a way that is much more efficient than having to read the papers in depth. So that's number 2. And the third one, it's actually a really good data analysis engine. So you could actually have it do analysis on data tables that you've generated in your experimental work. It'll actually write code or draw on code to generate graphs and figures and simple analyses. I wouldn't have it do like super sophisticated first time ever machine learning or anything, but simple analyses that you would need to get a data scientist to write for you. It can write the code and do the analysis for you. Those are 3 use cases that are huge accelerants.
Thank you. Let's take your question.
Thanks. Very exciting. It's amazing what all these things can do, but like everything else, it comes with a cost, right? I guess what my main concern is that in some places we've literally seen a doubling of electricity demand in 1 year. How do we keep this from making it impossible to control our need for fossil fuel use, our need for energy worldwide? It's going to consume every one of our best efforts unless we come up with a plan. So I would love to hear what you guys have as a plan.
I think Mike from NVIDIA and Pushmeet from Google DeepMind can speak to this. Mike, perhaps you want to start with your climate interests?
Well,
Putting you on the spot here.
It's not an easy question.
It's difficult to speak to the issue, which I think is about global industrial exponentiation of demand far beyond the climate use case. I think, making simulations more performant through using AI as a solver that might be more energy efficient than conventional PDE solvers is attractive. But of course, in climate, as you know, we'd rather simulate more than we can now as well. So that's not really a solution to your dilemma. Yeah, I don't have a good answer for you, Susan.
So I think just to sort of mention, it is a very important issue. And the amount of training and compute that goes into training the largest large language models is quite large. But people sort of recognize that it is a very important issue and are working on making that more efficient. Also, another point that I also wanted to mention is specifically in the context of science. For the most advanced science models, the amount of compute that is required is many, many orders of magnitude lower than what we are sort of thinking for LLMs. So even the most sophisticated biology model today, like the AlphaFold of the world, the AlphaFold 2 and even the next generation, the amount of compute that is used for training them is like many orders of magnitude less compared to even training a standard large-scale large language model. So we should all be we should use this if we think that the scientific benefit is there.
Well, thanks for thinking about it.
Yes, and I'd like to make 2 points too. One of the reasons I mentioned the interest in small data and small compute is because of the energy consumption of training one of these models. Another area where I think we need to pay more attention to is actually what I would call smarter algorithms or algorithmic changes, so that we just don't throw more data and throw more compute at solving a problem. Let's take your question.
Sure. Hi, Rick Silverman, Northwestern University. So as I understand it, AlphaFold uses the 200,000 or so structures as a training set. Structures are obtained under very non-physiological conditions, and if you change your conditions, you get a different structure, none of which really may match what the structure is in the cell. So the results we're getting from this AI are a structure that may be irrelevant because that's what the training set is. So what do we do with these structures?
Yeah, and that's, I'll take that. I think it's a very good question. And if you think about what AlphaFold is trying to solve, you're absolutely right that when scientists have found these structures by experimental means, they were structures in a specific context. And they deposited that structure in the PDP with that context, and maybe sometimes the context is missing. For example, they used a ligand to crystallize the protein and deposited that structure. What is very interesting is AlphaFold does not just use that information. It uses 2 sources of information. One is it uses evolutionary genetics and looks at what kinds of residues within the protein might be in contact because of evolutionary history. So there are many different elements that are going into it. When you don't give it the right context, it will in some sense default to the context that was used by the scientist who deposited that specific type of structure in the PDP. But what we have seen is that it still gives useful predictions, which are correct, which are useful, even when that context is not there.
Thank you.
Bruce Tabashnik, University of Arizona. Pushmeet, I love that you started with the goal is AI for the benefit of humankind. But what about AI that could be used very selfishly or even for evil?
What about it?
Are you concerned? What should we be doing? What kind of safeguards are needed?
Absolutely, and I think this is an incredibly important element, and that reasoning and that thinking has to happen at each step of the way. Before we start a project, we look at the ethical implication and the societal implications of the project. But even after that, even as the project is proceeding, for instance, let's take the case of AlphaFold. When we started, we thought this is a problem that the scientific community has been working on for a very long time, and given the feedback that we received from the scientific community, it was felt that this is a worthy problem to apply AI on. In the middle, when we looked at questions whether we should make the structures of, say, SARS-CoV-2 available to the scientific community, there was a question, are we adding to the noise at that time, or would this be genuinely useful? At a later stage, the question was whether now we have AlphaFold 2, how should we release it with the scientific to the scientific world? Should we open source it? Should we control access? In every stage of the process, we approached the scientific community. We looked at sort of experts in the field. We asked social scientists. We asked ethicists. We got feedback from them. What is the positive use case? What is the negative use case? And then sort of on balance basically made the right decision. But you are absolutely right. The purpose is to benefit humanity, and unfortunately sometimes technology can be used for malicious purposes, but our attempt is to make sure that there are safeguards at every step of the process, from initiation, in the middle, as the project is going, and then finally as the project concludes, that the beneficial uses are amplified and the malicious uses there are controls and mitigations in place to make sure that doesn't happen.
Thanks.
Sorry, Ben Neal, NYU School of Medicine. So first I want to thank all of you for this really great symposium, which I personally found really useful. My question is, several of you mentioned that AlphaFold was successful because you had 200,000 sequences matched to 200,000 structures, and then in the last talk we heard about the paucity of data for other models. But can you give us a map of how much data you need to solve, for example, the RNA structure program? How much data you need to solve the TCR peptide recognition, MAC peptide recognition problem? Because I don't know how to think about the scale of those problems.
Does anyone want to take that?
I mean, I think maybe you could get a ballpark estimate for some of those insofar as they bear resemblance in their complexity to the protein structure problem. But I get this question all the time, and that sort of presupposes you know the answer, right? In a sense, there's some like optimization landscape, and if that landscape's super rugged, you need that many more data examples. And we don't a priori know how rugged that landscape is or the complexity of the problem, but you could probably make some estimated guesses for RNA structure prediction from the protein structure prediction. I haven't done that, but I would speculate with great hesitancy that same order of magnitude might suffice. But
Daphne,
I think more generally it's really tricky.
Help out here?
So if I can jump in, I think it's important to recognize that a lot of the progress in modern day machine learning has actually come from data that's actually heterogeneous in the amount of labels that it has. So for example, I don't want to speak for Pushpin in terms of AlphaFold, but there's a lot of work that's been done on basically representation learning from data that's effectively unsupervised using techniques such as self-supervised learning. And then once you have that underlayer that is oftentimes giving you like a very concise and very semantically meaningful representation, you can sprinkle on top a much more modest amount of supervised training data, which is superpowered by having these foundation models. So for example, in the work that we've done in histopathology, we have about 100,000 patients worth, which is multi-hundreds of thousands of slides of the histopathology. We only had about 15,000 patients that had RNA-seq, but with a foundation model, it was trained on very broad-based pan-cancer, pan-tissue histopathology. Those 15,000 were actually enough to train that model that allows you to impute RNA-seq. And then on top of that, we had a very modest amount, even more modest, of things that correspond, for example, to patient response data. But with the RNA-seq model that was used to fine-tune the foundation model in histopath, you were able to kind of make use of that much more modest amount of training data. So when I think about your TCR peptide MHC recognition, I would start with a protein foundation model, which is, you could train on all proteins, and that gives you the language of proteins, so to speak. And now you can start to make use of smaller amounts of data that are much more problem-specific to kind of train a model that is geared at your specific prediction problem. I'm sure Pushmeet can speak even more.
Because I'm sure they're working on that problem.
Yeah, so I think this is a very, very important question. And I agreed with many of the comments that Jennifer sort of mentioned in the talk, but I disagreed with one. And the thing she made the comment that the phase transition of progress in AlphaFold was with ML and AI alone. AlphaFold did not happen with ML and AI alone. It was not a machine learning team that solved AlphaFold. We had a very multidisciplinary team that solved AlphaFold. There were biophysicists in it, there were structural biologists in it, There were a lot of biologists within the team. In machine learning, there is data that is introducing insight to the model. But there is another source, the inductive bias, the design of the architecture itself. So it's not a simple sort of question like how much data do you need to solve this problem, because it's also a function of how much inductive bias you have already baked into the architecture itself. If you have already given the model a lot of insights that have been developed in that field, then the amount of data that you need to solve that problem dramatically shrinks, right? So when DeepMind was working on AlphaFold, every single team had access to the same amount of data. It was not just a function of data. It was basically the amount of insights and the amount of data and the amount of engineering and the machine learning sort of knowledge that together went in to solve the problem. Now there are systematic analysis that we do, and there are scaling laws to understand for a specific architectural choice with a specific inductive bias. You can sort of evaluate if you can do a sort of ablation analysis and say what is the generalization performance with this data, this amount of data, this amount of data, this amount of data, and it will give you some trends. And if you many times you can actually extrapolate those trends and they turn out to be good, right? But that whole sort of study, that scientific study, is in its very early stages. And a lot of research needs to be done in terms of can you really predict the information sort of content that needs to be in your training set to be able to achieve a certain level of generalization performance.
Thank you.
Thank you.
Yes, again, I'll also thank all the speakers. I'm Bill McKinnon. I'm at Washington University in St. Louis, and I'd actually like to go back to almost the very first thing you said, Dr. Wig, in your history tour. The Turing test was passed by ChatGPT. And even if we think of ChatGPT as an army of well-intentioned but not necessarily well-informed undergraduates, no, I think this audience would not consider that army to be conscious in this, at least in a conventional sense. So is machine learning, AI, is it actually working in a way, or is it actually replicating actual neural, biological activity, or are we merely algorithms, and is general intelligence and consciousness an emergent phenomenon that is not there yet?
Start.
And I know you've all thought about this kind of thing.
No. I think, when ChatGPT, and it's like basically made the Turing test moot in some sense, yeah. I think it re-raised the question of what is intelligence, and all sorts of philosophical questions, like the difference between machine intelligence and human intelligence, and the kind of intelligence when you have humans and machines working together, and what kinds of problems can be solved by those kinds of entities that cannot be solved by machine alone or human alone. What is consciousness? All these sorts of philosophical, scientific questions are actually being raised. And I think one of the papers that I personally thought was trying to raise this question was the Sparks of AGI paper that was put out by my Microsoft colleagues, and it really raised the question of what is intelligence? But does any of the AI experts on the panel want to address this question, or the philosophy behind it? No, okay.
I think you summarized it well, Jen.
Okay,
Jenny, I'd share an anecdote.
Yeah, go ahead, Mike.
This is just an anecdote. I was talking to a friend at the Columbia Leap Center, a computer scientist who was passionate about trying to understand more detail about how machine learning was solving complex analysis tasks by asking the machine learning to write code that was human interpretable and therefore understand the sequence of actions that was going into a solution. And the anecdote he told me was cutting and pasting 2 columns of Excel spreadsheets, X and Y values corresponding to a mathematical function, giving that to the AI and being like, what's the mathematical function? And in looking at the generated code, it was clear that the AI was choosing to visualize the function, use its image recognition to recognize the solution was cyclic, and start guessing trigonometric possibilities. And that felt, relatable. I don't know if that relates to your question or not. Also mimicry, perhaps, but...
Another point, to take your question about neuroscience a little deeper, there's actually a new National Science Foundation, National AI Institute, led at Columbia on AI and neuroscience. And they are actually looking at deep neural networks, by its very name, of course, was inspired by building a computer that connects, processors together like your brain is connected. And, it's just a metaphor. It's not, if you look at a neural network, they're very simplistic with respect to how our brain is actually wired. So NAI Center on AI and Neuroscience will look in both directions. How can AI, of course, with data, help us understand the brain better? But also, are there other analogies of how our brain operates, is circuit up, wired up, and so on, that can suggest new kinds of AI models? So it's, again, a little philosophical, but right now, please remember that deep neural networks were inspired by what our brains look like.
Well, certainly, I think maybe in a way, what I've learned from these talks is that in a way that they do emulate how people actually think, trying to just input all the data they've acquired in their lives or been taught as to what the outcome will be, what will happen next, with the weather or climate. And it's not science in the sense, it's not physics-based.
So that's another, I'm sorry to speak, but since I have the microphone, I will. That's another point I wanted to make, which no one has made yet, and that is, as much as we are enamored with the neural networks, the technology of AI, generative AI, and so on, all those big fancy diagrams that people are drawing, we actually don't have a scientific understanding of why they work. Right now, it's an experimental science. We have a lot of data, we train a lot of models, we do a lot of things with those models, and it's all amazing.
But Jeanette, I would argue that we also don't understand that about the brain either.
That's right. There are a lot of things that we don't have theoretical scientific understandings of, but kind of work in nature, in practice, and they're amazing, and sometimes they help us solve really challenging problems. Next.
David Weinberg, Ohio State University. My question is connected to the last three talks, and so I understand the impossibility of using data that you generate to train your model, but my question, in the climate context, can you use a physics-based model to perturb things and generate new data using that physics information that then actually add to your training set? And in the drug discovery context, Daphne used the term imputation a number of times, and I felt like I almost understood what that meant, but it also seems a little bit like this, generating your own data from the model. So I wanted to ask if you could be more clear on what imputation means and why it doesn't fall under this paradox.
Yes, let's start with Daphne.
Yeah, no, thank you for asking that, because when Jennifer presented her talk, I wanted to clarify the difference. I think Jennifer was alluding to an entirely self-contained model where it just feeds upon itself and hallucinates more and more data and uses it to train itself. And that is absolutely a black hole of nonsense that one should avoid at all costs. What we're doing is actually taking data that has been measured, that has a reality to it, and then using a model that was trained on other data instances to complete, if you will, the unmeasured components. And so, first of all, it starts with a model that was trained on not this data set, and second, it uses a substrate of reality from which additional layers can be completed. So I think that is very different than an entirely self-contained system that just hallucinates on its own. And I think it's a very important distinction.
Just to continue on that
Yes,
sort of point, one of the projects that I mentioned on graph neural networks for material discovery takes exactly the approach that you are sort of suggesting. It takes the active learning-based approach where it says, I want to understand what materials would be stable. In order for me to do that reasoning, I first learn from some data of quantum chemical simulation that has been done at scale, but you cannot sort of compute, you cannot do those calculations exhaustively for all systems.
Yes.
So what the model says is that I'm particularly uncertain about these systems. Can you run many more quantum chemical calculations on these specific systems and add them back to my training set? And that sort of virtuous loop gives you a very high level of performance, which was behind that breakthrough in terms of material discovery stability prediction.
And maybe just to add one other important thing, when you do this kind of imputation or data generation or whatever, it is very important to have a held out data set that was not used as part of your training to make sure your predictions actually represent reality faithfully. And we absolutely do that, and I know others do as well. So I think that is another component of the avoiding the hallucinated data trap.
I have been given the hook, so therefore we need to stop, but I believe the panelists will be around for a while, and the people who were not able to ask questions, please feel free to come up and ask us your questions.